People still getting drinks, but... Okay, good evening everyone. My name's Antoinette. I've got Ellen at the back there waiting for some of the others coming through. But before we need to obviously start getting the ball rolling. So thank you so much for joining us this evening and welcome to the third Raising the Bar Adelaide event. We're very, very excited to bring Dr. Hannah Wardill, yep, to get that right. And she's going to be talking to us about, and apologies for those people that are eating at the moment, chemotherapy with a side of poo thanks. Those of you who have just joined us by being here tonight, this is the Raising the Bar Adelaide, which initially started in New York with the intention to turn pubs into classrooms by combining education and pop culture. So, this event has been around the world, so it's been around in Hong Kong, London, Auckland, Sydney, and now the city of Norwood, Payneham and St. Peter's, and we're very proud for it to be here in Adelaide again. So, as we speak, or as I speak to you right now, there are 9 other pubs around the city tonight that are all actually hosting the talks by academics and esteemed speakers. We really hope you enjoy it tonight. And please don't forget to share any of your experiences and pictures via our social media accounts with the hashtag RTBADL, as you can see up the top here as well. Please, we do look forward to your creative posts if you do put them up. And please be respectful of our speaker tonight and enjoy Hannah as she takes on board. Thank you.
Okay, thank you very much. I don't love speaking into a microphone, so hopefully you can all hear me well enough and it's clear, and luckily we've got the tech person who will keep on top of that if any issues arise. It's really lovely to actually speak to a room of people tonight. You know, for the last few months we've all been having meetings and conferences via computers and Zoom and all of that, so it is really nice to speak to real people tonight, although I am a little bit nervous. So please forgive me if I have any mental breakdowns throughout this session tonight, but hopefully not. Okay, so to get the room a little bit warm, I want to get you to all have a think about the last time that you went to the toilet. And I'm not talking about number ones, I'm talking about those slightly more embarrassing, and sorry Sharon, I know you're eating a mushroom burger, but number 2s. So let's just call it poo, let's get it out there in the open, get comfortable with the word, because for the next 45 minutes, or maybe slightly less, we'll be talking about poo. So we need to be comfortable with this terminology. Now, I'm assuming that for most of you, the last time you went to the toilet, maybe it was this morning, hopefully it's fairly regular. You probably sat down, you did what you had to do. There's a few millennials in the room, so perhaps you scrolled on your phone for a little while. Maybe you were at work, so you sat there a little bit longer, because getting paid to poo is quite a great novelty, I think. But ultimately, I think you probably finished up, flushed, washed your hands, hopefully, you know, COVID, stay safe, and you didn't think much else of it. But what about if I told you that every time you go to the toilet, you actually become a little bit more human? And I know it sounds kind of ridiculous, and you're probably thinking, who is this weird girl they've got to talk to us tonight? But bear with me and I will explain. So the human body is colonised with a rich ecosystem of bacteria and other microorganisms, and we call this our microbiome. And actually the human body is home to over 100 trillion different types of microorganisms that colonise every surface, every nook and cranny of our body. And so each and every time we go to the toilet, we lose some of these microbes. And if you look at the human body, we know that for every human cell, there are 1.3 times the number of microbial cells. So if you look at a human genetically and you take into consideration their microbial community, we're actually considered much more microbe than we are man. And so each and every time we go to the toilet and we lose some of those bacteria into the toilet, we actually shift this balance ever so slightly into the favour of being more human. And it's a little bit of a stupid anecdote, I suppose, that I use to get people to think about all the vast microorganisms that call our body home. But I think it's a really nice way to emphasise just how many bacteria, viruses and fungi we have all over our body. Now, the vast majority of these microbes, while they colonise all aspects of our body, are largely found within our gastrointestinal tract or our gut. So for those of you that might typically associate the gut with just the stomach, it's actually anywhere between your mouth and the other end. So this means mouth, esophagus, stomach, small intestine and large intestine. And the vast majority of our microbes really like to hang out in our large intestine or in our colon. And in the average human adult, there are actually over 2 kilos of bacteria that are found within our gut. And I think this is a pretty profound statement. I mean, 2 kilos is a huge amount of bacterial product. It's like 3 bottles of wine, maybe. And for me, as a fundamental scientist with an interest in infectious diseases and microbiology, this really astounds me, because us humans are made, or are physiologically made, to fight off infections, pathogens and bacteria, just like the ones that colonise our gut. So this raises a really important question, I think, as to how we tolerate these microbes, or perhaps more importantly, why we tolerate these microbes. I mean, we've got 2 kilos of them in our gut. We must tolerate them for a particular reason. And what we're starting to understand is that it actually comes down to evolution. So some of the earliest living organisms actually buddied up with a little microbial sidekick, I suppose, in order to have a competitive edge over their peers or over their friends. Now, this bacteria or whatever microorganism that this organism buddied up with, it carried out a particular function that organism could not do alone. However, it's really important to realise that these bacteria aren't just these lovely philanthropic volunteers. They don't do this for nothing. They really do expect something in return. And so in many circumstances, these organisms would return or would reward their bacterial buddy with some type of present or nutrient or something that would keep them sticking around. Otherwise, they're gonna bugger off. So this type of relationship where 2 seemingly unrelated factors or organisms come together to have mutually beneficial responsibilities and outcomes is what we call mutualism, or a symbiotic relationship. And there are many symbiotic relationships that exist in the natural world. And I think probably the clearest example, or perhaps something that each of us can resonate with, is the relationship between a flower and its pollinator, so a blossom and a bee. And of course, without each other, the flower couldn't go on to turn into a fruit, and that bee wouldn't get the nutrients from the nectar that it requires to live. Now, just like that flower would potentially die without its little bee, us humans would really struggle to survive without the complex ecosystem of gut bacteria that we have. And the way in which we started to understand this was by using a specially engineered type of mouse, which is called a germ-free mouse. And as their name suggests, these mice have absolutely no bacteria on them or in them. They're completely sterile. And scientists are starting to use these germ-free mice to try and understand or learn exactly how our gut microbiome and other microbial populations in the body contribute to certain diseases and also to other physiological factors and events related to health. However, one of the first things that we identified or learnt by using these germ-free mice is that actually they're very difficult to keep alive, and they die very quickly if we leave them in a normal environment. And what we found is that the only way we can keep them alive is by keeping them in very sterile situations where temperature, air pressure, food and water are all very, very strictly regulated and extremely sterile. And so I guess you could sort of compare it to the boy in the bubble, where everything is really, really carefully regulated in order to prevent that very vulnerable mouse from succumbing to simple challenges. And so unless we all feel like having a future where potentially we're men and women in bubbles, it's probably time that we started thinking a little bit more carefully about our gut bacteria and how we can better support these gut bacteria. But before we get into that, I think it's really important that we understand exactly what our gut bacteria do for us. So us scientists, we're slowly starting to scratch the surface of this relationship between humans and their gut microbiome or their gut bacteria. And we're starting to understand that they actually contribute to a myriad of different physiological factors and also disease mechanisms. But I think probably the factor that is best understood for the microbiome is its ability to digest and break down dietary fiber. Now, fibre is something that I think we're all aware of. We're all told to eat a lot of fibre. It's part of a healthy, nutritious diet. We get it from fruit, we get it from vegetables and whole grains. But actually, us humans are completely incapable of breaking down and digesting fibre. But luckily for us, our gut bacteria, they're absolute powerhouses at it. They're phenomenal, and we only need to look at the gut bacteria of termites to understand just how good they are at breaking down very fibrous substances. I mean, termites live almost exclusively on a diet of wood, and these termites still thrive, and this is simply because the gut bacteria that they have and we have are very good at breaking down dietary fibre. Now, when they break down this fibre, they liberate a small portion of energy from that dietary fibre that we would usually be unable to identify or capture alone. But as I mentioned before, these gut bacteria, they need something in return. They're not volunteers, they want something, they want to be paid for their work. And so the human body has come up with a pretty phenomenal way of returning or rewarding our gut bacteria for the hard work that they do in digesting our fibre. And we've done this by recycling the waste products, or I guess kind of the poo of bacteria. It's like poo in poo in poo. It's amazing. So when bacteria gobble up and start to digest our dietary fibre, they produce their own waste product, and these waste products are called short-chain fatty acids. Now, these short-chain fatty acids are extremely potent nutrient sources for the cells that line the inside of our gastrointestinal tract, so the cells that line our gut, and we call these cells epithelial cells. And what these short-chain fatty acids do is that they stimulate these cells to grow, they help them overcome any injury or stress, and most importantly, they stimulate the production of mucus. Now, this last point is really important because bacteria honestly love nothing more than mucus. They love to feed on mucus. And so in return for digesting our dietary fibre, we reward our gut bacteria with a warm, cosy, climate-controlled environment with a constant supply of nutrients. And this is the absolute essence of that symbiotic relationship that exists between us and our gut bacteria. Now, as we begin to learn more and more about our gut bacteria and the weird and wonderful ways in which they support human health, what we're starting to understand is that actually, our gut bacteria do much more than just affect their local environment, so just the cells in the gastrointestinal tract. And we're starting to understand now that they actually have profound influences on things like our immune system, our endocrine system, and even our central nervous system. So they're able to regulate things like mood and our risk of depression and anxiety and things like that. And so this has really started to see a boom in research linking sort of disrupted or altered gut bacteria with certain diseases. And I mean, this area is growing at a rate of knots. It's almost impossible to keep on top of the literature. But we do know that changes in our gut bacteria are now linked with, they don't necessarily cause, but they're linked with certain diseases like cancer, inflammatory bowel disease, obesity, metabolic syndrome, autism, Parkinson's. I mean, the list is honestly endless, and it feels like it's consistently growing. It's a huge area of research at the moment. And while this has certainly led to some major advances in our ability to treat these diseases and understand these diseases, it's also really opened up the door for commercial exploitation. And I think that this has probably been the most well articulated or demonstrated in the case of probiotics. So I'm going to do a bit of an awkward thing, but a quick show of hands of how many people have maybe tried a probiotic at some stage as a bit of a quick fix for an upset stomach. Okay, a lot. And yeah, wow. I'm not actually too surprised by that sort of response, because at the moment the probiotic market is estimated to be at about $50 billion US. I mean, it is a huge part of the health and medical research commercialisation aspect. And by 2027, it's estimated to reach $95 billion US. It is a huge market. And the probiotic companies have really done a fantastic job of, I suppose, jumping on board or leveraging on our obsession with gut health and gut bacteria. And while this is great, you know, they're making claims that they're going to be supporting immune health, supporting brain health, supporting gut health. What we actually know is that there have been over 500 rejected health claims for probiotic companies in Europe alone. And this is a bit of a major problem, because what it means is that probiotics are really a little bit ahead of where the literature is and where the science is at the moment. And this is certainly not to say that probiotics have no space in science and in medicine, because there are certainly examples of where probiotics have had profound benefits. For example, in necrotizing enterocolitis, which is a disease of the gut in children, there have been really positive results for probiotics. But the problem with probiotics is that they somewhat oversimplify our understanding of the gut microbiome. And so if you take away one thing from today's presentation, it's that diversity is absolutely critical in our gut microbiome. There are hundreds of different types of bacteria, and it's really important that we support all of these diverse and wonderful bacteria within our gut. Now, I'm going to demonstrate this point with an analogy of a rainforest. So if you think of a rainforest that is made up of just one type of tree, a monoculture, if there was a disease that came in and affected that particular tree, you can imagine that that disease would jump from tree to tree to tree very quickly. And it would very quickly have a devastating impact on the composition and the health of that rainforest. Now, if you imagine now a very diverse rainforest with many different types of trees, bushes, vines, it would attract insects and birds, you can imagine that that disease would have a very tough time jumping from tree to tree. And very quickly, the diversity of that rainforest would simply suffocate that disease and would result in the rainforest maintaining a healthy or diverse state. Now, this is exactly the same as what happens in our gut microbiome. There are hundreds of different types of bacteria, good and bad, that all live in harmony, where the good bacteria help to support each other, and in doing so, they squash or they suffocate out those bad bacteria, and they maintain them at a level that is tolerable for the host, for us. And this is ultimately the key function or the key attribute of our gut microbiome that makes it so profound and so strongly linked with health and disease, is because its diversity is critical in maintaining the vast variety of functions that it creates. Now, just going to take a quick drink. Okay, so far we've learnt that the microbiome is this diverse ecosystem of bacteria, largely bacteria, but also viruses and fungi that inhabits all aspects of our body. But I think one of the most interesting aspects about the gut microbiome is that actually each and every one of us has a completely unique composition of gut bacteria. So, you know, your bacteria are going to be different to your bacteria, and what we're starting to understand is that this is so unique that it's been likened to that of a fingerprint. So it's extremely unique. And this is because there are a myriad of factors that influence the composition of the microbiome, most of which are completely unique to us. So for example, let's take genetics, something that we can all probably attribute to being unique to all of us and something that makes us individuals. And if we look at genetics in terms of the gut microbiome, we know that genetics do play a role in shaping the types of bacteria that we find in people's guts. And we've learnt this by looking at the gut microbiomes of twins. And we know that those twins do have a strong core set of bacteria that are common and consistent between them. However, if you take those twins and you allow them to grow up in different environments, I will almost guarantee that those twins will go on to develop completely different gut bacteria. They'll have different amounts, different compositions, different balances between good and bad. And that's because our gut bacteria actually is much more influenced by the environment that we're in rather than by our genetics. And so what we actually know is that these environmental factors start from the very first day that we're born. Each and every one of us in this room are born under fairly sterile situations. We're born with very few gut bacteria. And what happens in the process of childbirth is that there's actually direct transfer of bacteria from the mother to the child, and we call this vertical transfer. Now, if we compare the gut bacteria or the gut microbiome composition of children born via vaginal delivery versus caesarean section like me, we know that they have vastly different gut bacteria all the way through to adulthood. And actually, if you look at the gut bacteria of children born by caesarean section, we know that they typically have more bacteria that we would usually associate with surfaces of the skin or even surfaces in a hospital. Now, this is not to say that children born via caesarean section have worse or bad microbiomes. It's just that they have differences in the types of bacteria found within their gut. And what we know is that these environmental factors don't just stop there. I mean, children that were breastfed versus formula fed, they have differences in their microbiome. Birthday time over there. And also, you know, there are a myriad of factors in our early life that affect its composition. So if you have pets, if you grow up with siblings, all of these things affect the composition of our gut bacteria, and they are ultimately the reason why each and every one of us have a completely unique composition of gut bacteria or our microbiome. Now, I think this point of having this unique microbiome is something that really seems to fascinate a lot of people, and I was actually talking to Henry the other day, my friend who's in the audience, and he said, oh, well, so if everyone has a unique microbiome, does that mean you could technically catch a potential criminal if they left a cheeky poo at a crime scene? And while I would have loved to have said yes, unfortunately, just that is not the case. And this is because in addition to being extremely unique, our gut microbiome is extremely plastic. And this means that it's able to undergo very rapid and transient and dynamic changes in response to a lot of, or a variety of different environmental factors. So I mentioned before, all of those factors related to birth and early life exposure and experiences shapes our microbiome. But what we also know is that things in our adult life also affect its composition. So things like stress, diet, I mean, if you move house, if you eat a spicy meal, if you drink too much alcohol, if you move house, all of these things are known to affect the composition of our gut bacteria. And this is ultimately why we have differences from day to day in the types of bacteria that we find within our gut. So unfortunately, we can't be catching criminals based on poo, but it's certainly something that I'd like to look into. So I'm just going to quickly pause there. I realise that I do have a tendency to speak fairly quickly, and this is an area where there is a lot of information. So I would like to now throw it over to you guys to see if you have any questions or perhaps areas that I may have glossed over too quickly, or any questions from this point? Yes.
Testing.
You mentioned studying twins, but you also said that siblings growing up in the same family can have different microbiomes. If they have the same diet predominantly, what is it that causes the differences?
Absolutely. It's a really good question, and I think it's something that we're slowly starting to unravel. It's kind of the same point of nature and nurture, I suppose. And diet probably has the largest influence on our gut microbiome. So certainly if these children were being fed the same things, it's very likely that they would have quite similar gut bacteria in the composition of their gut microbiome. But of course there are a lot of sort of psychosocial aspects as well that can shape our gut microbiome. So if children are experiencing different levels of stress at school, or perhaps they're exposed to different stimuli from peers in their classroom, these things can also affect the composition of the microbiome.
I'm interested in how stress affects your body, and I notice particularly that if you get sleep deprived and you tend to eat all bad foods and stuff. So is there much work being done on that to correlate stress and that sort of thing with the body?
Yes, absolutely. So I think, when we think about the relationship between stress and the microbiome, what we start to think about is this gut-brain or brain-gut axis. And I think we can probably all relate to that in some way because, of course, get a little bit nervous, you want to go to the toilet, right? Or get a bit nervous and you have butterflies. This really does emphasise that there is quite a lot of communication between our brain and our gut. And so I think we've probably learnt the most about this sort of aspect in people with irritable bowel syndrome, which to this point has really been considered a major or a gastrointestinal disease because people present with diarrhoea or with constipation. But actually, if you look at their gut is pretty normal. Like there's no evidence of disease, no evidence of any injury to the gut. And so we're now starting to think that actually IBS is maybe more of a neurological disease that is brought on by some degree of stress. So it's really important to note, I suppose, that while we're starting to see these relationships come out, we really don't know sort of cause and effect. And in reality, there's probably going to be a bit of bi-directional communication where stress affects the microbiome, but then of course it affects your interest in eating and it affects your sleep, which then has this confounding effect on the microbiome. So it's a very difficult thing to tease apart, but certainly stress is documented to change the composition of your gut bacteria. One more?
So you said everybody's got different compositions, different concentrations, different populations, but do you know enough to say that it'd be good if I had a little bit of that one, some of that, minimum of this one?
Yeah, that would be pretty fantastic. And I guess as well, you're starting to touch a little bit on this idea that people are really interested in the types of bacteria that they have in their gut and what that might mean for their health and potentially their risk of disease. And, there are these tests that you can order online that are like these poo tests, and you send it off and they sequence the types of bacteria that are present in your poo, and they try and make predictions about your risk of diseases. But I think those types of attempts are a little bit beyond where the science is at the moment. Certainly we have some indicators of how diverse your microbiome should be and the general balance between good and bad, but I don't think we're quite at the point of saying, you should have this and you should have that. And because, you can have very diverse gut bacteria in 10 different people and they're all still relatively healthy. So there's probably a threshold within that space that we're still not quite certain of where those values lie. Yep. Shall we move on in the interest of time, or have we got... Oh, one more. Hello.
There's a bit of a fad for fermented foods at the moment, kimchis and these sort of things. Does that carry much benefits?
Yeah, it's a bit of a tricky question. So the probiotic sort of angle that I was pushing before was very much around commercial probiotic, pills and capsules and things, because they're typically made of just one gut bacteria that's associated with some, status of health, a bifidobacteria or a lactobacillus. And so what that does is creates a microbiome that's dominated by one type of good bacteria. In the case of fermented foods and yogurts and things like that, the diversity of bacteria found in those products and in those foods is much higher than what we see in commercially available probiotics. So I think in a generally healthy person, there's certainly no harm in eating those foods. there may be some benefits, but I think ultimately those benefits are probably pretty similar to if you eat a well-balanced, plant-based, high-fibre diet. So certainly no harm, but potentially not the profound level of benefit that maybe we're seeing in the literature and in the public space at the moment. Yeah. All right. Let's move on then. Thank you for those questions. They were great. All right, so far we have touched on the fact that we have this amazing ecosystem of bacteria that calls our gut home, and we have this balance of good and bad bacteria that can be influenced by a myriad of different things. We've already touched on the point that each of us have a unique gut microbiome, and as we started to touch on, this has really got the scientific and the medical community very interested in what exactly this might mean in terms of dictating an individual's risk of disease, or perhaps our ability to predict how an individual will respond to a certain intervention or drug or medication. And I think that this latter point has probably been really well articulated or demonstrated in the field of dieting and weight loss, something that I think all of us have probably, engaged with at some point in our lives. And we probably all have that one friend who eats whatever they want and never ever puts on weight. Or perhaps you just have to watch one episode of The Biggest Loser to understand that not everyone responds the same to a low-carb diet. I mean, some of them go on to lose like 10 kilos in a week and other people can't even lose one. And this is because we all respond very differently to dietary intervention. And this heterogeneity in people's responses to diet has really got a lot of people in the weight loss and dieting and obesity field very, very interested. And there's a group in Israel that have been particularly prolific in this research space. And they really wanted to understand what exactly it is about people that makes them respond well or not so well to dietary intervention. So what they did was they got a large group of obese individuals and they put them all on the same diet. They then recorded how these individuals responded to this diet in terms of weight loss and other indicators like blood glucose. Now, what they were able to show was that if they looked at the types of gut bacteria that were found in these people's poo before they started their diet, they were able to show that the people that responded well to the diet, so those that lost weight, tended to have very different gut bacteria to the people that responded poorly to the diet. Now, what they were able to do next in a completely new group of people was that they were able to just look at the composition of their microbiome, and they were then able to predict how those individuals were going to respond to that diet. So they could identify the responders and they could identify the non-responders, only based on the composition of their gut microbiome. Now, this group, they're very ambitious. They took it even one step further, and what they were able to do after this in a completely new group of people again was that they were then able to look at their gut microbiome and match the individuals to a specific dietary intervention, whether it be high protein, high plant-based, high fibre, low carb, many, many different combinations of diets. And they were able to show that if they did this in this way and they used artificial intelligence to predict which diet would be best, they could optimise weight loss in a manner that actually outperformed the recommendations of a dietitian. And I think we can all agree that this is a pretty profound finding. It's very much highlighting that we can personalise the way in which people respond to certain dietary interventions, and we can optimise the results for every individual based on the composition of their gut bacteria. And this is exactly what I'm hoping to do in the field of cancer therapy, because just like diets, we know that people respond very, very differently to various chemotherapies and other cancer agents. And we know that this is particularly the case in terms of the types of side effects that they go on to develop. Now, I'm going to assume that at least a few of you, maybe not that many of you, hopefully, are thinking, oh, side effects, I mean, are they really that much of a problem? Like a little bit of hair loss, a little bit of nausea, it's not that bad, is it? If the person's cured, suck it up. Well, I say wrong. I say wrong because I think that, I don't say it to be mean. I say it because over my research career, I've had the opportunity to work alongside some fantastic patient advocates and consumer advocates, some of whom have joined us here tonight. And these people, they have had either directly or indirectly through family and friends, they've been exposed to the challenges of cancer. And they have a really unique perspective on the barriers or the challenges that are faced by people going through cancer therapy. And they also have a really unique perspective on the types of unmet needs in people's cancer care. Now, what I've come to learn from working alongside some of these advocates is that the side effects of cancer therapy actually have a major impact on their quality of life, both while they're going through cancer therapy and even afterwards. Additionally, we also know that these side effects can have a major impact on their ability and also their willingness to carry on with treatment, because we know that when these side effects get bad, the first thing that the oncologist will do is either reduce the dose of their chemotherapy, perhaps delay the next cycle, or in severe cases, they'll completely stop the chemotherapy. And so for these people, their side effects actually prevent them from being treated in the way in which they should be. What we also know is that it doesn't just end when the cancer ends. 1 in 3 survivors of adult cancer will go on to develop a chronic physical or psychosocial or even financial complication that is directly related to their cancer therapy. So just to put things into perspective, and I don't want to bring the mood of the room down too much, but it is a really good statistic to emphasise the point that I'm trying to make. So children that are diagnosed with blood cancer today, a child diagnosed with blood cancer today, has a greater risk of dying due to the complications of their treatment than from the disease itself. And I think we can all agree that this is a pretty staggering statistic, and it's certainly an extreme example. But what it emphasises is that while our ability to treat cancer has grown enormously, I mean, children are almost, I think the survival rates for childhood cancer are about 90% now. It's pretty high. And so we've really, learnt a lot and grown enormously in our ability to treat cancer. But unfortunately, our ability to support these people through intensive and increasingly challenging cancer therapies just has not advanced at the same rate. And unfortunately, we still remain in this mentality, I suppose, where side effects are seen as a necessary evil in order to achieve a cure, or they're just something that shouldn't be spoken about. And so this was really one of the key motivating factors that helped me decide to start my own research group called the Supportive Oncology Research Group. And I really wanted to use my lab as an opportunity to increase the visibility of side effect management and the importance of symptom management in people going through cancer therapy, give patients and advocates a platform to highlight the areas of unmet need or areas where they would like to see more research, and also to develop new and innovative ways of better supporting people while they're going through cancer therapy. Now, one of the biggest challenges that we face in the field of supportive oncology is just like those diets, everyone responds very differently to cancer therapy in terms of the side effects that they go on to develop. Quick water break. So we can have 2 very similar looking people, same age, same sex, same cancer diagnosis, and very similar treatments. But what we know is that one person may go on to develop a raft of potential complications, so nausea, vomiting, diarrhoea, infection, nerve injury, memory loss. And the other person will go through cancer therapy seemingly unscathed. Yet we have absolutely no idea why this is the case. And this is a major problem because it means we have no ability to predict how an individual will respond and therefore prepare them for what they're likely to experience throughout their cancer therapy. And I think we can all agree that this level of uncertainty, it's certainly anxiety inducing. I mean, tonight I was anxious for the whole week because I had no idea who was going to be in the room, whether you'd be engaged in the content, whether there'd be people singing happy birthday all night. I was really nervous. And so I can only imagine that these people going through cancer therapy are experiencing extreme anxiety from this lack of knowledge about what they're likely to experience. And I think that This was really nicely articulated in a recent report from the Macmillan Foundation, which is a cancer care organisation in the UK. And they have a very strong consumer advocacy element to their reporting and to their work. And so this report was very much focused on patient experiences, anecdotes, testimonials. And obviously today I don't have time to go through each and every one of these, but what I want to emphasise is that this report was really littered with language like, if I had known, or I wish I had known, or it was a shock to find out later. And so what I was able to conclude from this report is that for many people going through cancer therapy, the side effects are really quite unexpected, and they have a heavy and unexpected cost to their quality of life. And so to tackle this problem, my lab has started a project called the PREDICT study, and as its name suggests, it's aiming to predict how people will respond to cancer therapy, both in terms of how effective their cancer drug is at killing the tumour, as well as the side effects that that individual is likely to experience. And so what we're doing is we're collecting poo samples from people before they go through chemotherapy, and we're looking at the types of bacteria found within that sample to understand what bacteria are found in that person's gut. We're then looking at and measuring the different types of side effects that these patients go on to develop, and we're trying to develop predictive models that will allow us to predict those certain side effects based on the composition of the gut microbiome. Now, what we've already been able to show is that people that have higher levels of a particular bacterial cluster called Blautia, they tend to do better in their subsequent chemotherapy round, so they're less likely to go on and develop diarrhoea. We've now isolated this particular bacterium, and we're looking at its behaviour in the lab to try and understand exactly why it may be responsible for this finding, and perhaps if there are ways that we could stimulate its growth in people before they receive chemotherapy, or perhaps even look at administering this particular bacteria at the same time that we give chemotherapy. But what I want to emphasise is that this research is very much in its infancy. We've only looked at this in a relatively small number of people. But ultimately what our goal is to provide these people with much more clarity around the types of side effects that they're likely to develop throughout their cancer therapy, so that people can ultimately make much more informed decisions about their treatment. Now, I think we can probably all agree that this is particularly pertinent in end-of-life care, where quality of life is certainly prioritised over quantity of life. But I want to argue that I think that this level of insight is relevant to all people with cancer, because we all want to know the outcomes of our treatment. And of course, predicting how someone, or predicting an individual's risk of certain side effects, will allow that individual to adopt particular behaviours proactively to hopefully minimise the impact of those complications. However, I think really predicting how someone will respond is only part of the problem, I suppose. And so what I hope to do is, just like the diet study, I want to use this information in a way that we can match the right treatment to the right individual based on the composition of their gut microbiome, so that we can personalise their treatment, and we can personalise it in a way that meets the exact needs of that individual. So we would look to be enhancing the efficacy or the effectiveness of their cancer therapy, whilst also making sure that it was minimally toxic, so less side effects, particularly those that were deemed particularly impactful for that individual. Because of course it's really important that we also take into account the individual's perceptions and their own specific needs. So this might mean for someone who really loves playing the piano, that we would be able to avoid therapies that would cause really damaging nerve injury in their hands. Or perhaps for the singer, it would mean avoiding treatments that had particularly troublesome and painful mouth injury and sores in their mouth. You know, it's about tailoring treatment so that we can match the exact needs of the individual. But of course, like I said, predicting how someone will respond is only part of the problem, and we really do need to find ways in which we can prevent certain side effects from occurring. And so I would like to spend the last part of this presentation highlighting a new study that we're about to start at the Royal Adelaide Hospital in people with blood cancer. So I mentioned before that there are a myriad of factors that can affect the composition of the gut microbiome, and these can affect it positively, like a high fibre meal, or negatively, like stress. And unfortunately, two of the factors or the things that we know have pretty bad influences on the composition of our gut bacteria are chemotherapy and antibiotics, both of which are readily used in the treatment of blood cancer. Now, from studying the poo of people undergoing chemotherapy for blood cancer, we've found that they have profound changes in the composition of their gut microbiome. So they have major decreases in the diversity of their gut bacteria, and thinking back to the rainforest analogy, we can all understand, hopefully, that this leaves an opportunity and space for the bad bacteria in their gut to flourish. And so what we know happens in people with blood cancer is that they actually have really big increases in the amount of bad bacteria in their gut. So we see a lot of E. coli and Enterococcus. And what we're starting to understand is that these changes, or these bad bacteria, actually are really important in some of the side effects that these people go on to develop. So things like diarrhea, infection, and also other particularly challenging complications like graft versus host disease. But what we're starting to understand as well is that actually the longevity of these changes can affect things long after the person's cancer has been treated. So we know that people with particularly chronic changes in their gut microbiome are more likely to experience fear and anxiety about their cancer potentially returning. We also know that the changes in these gut bacteria may even affect things like relapse. And so I think we can all agree that there is a very clear need to find better ways to help support these people's gut bacteria, or at least restore them after they've finished chemotherapy, so that we can potentially mitigate some of these complications. Making a mess, sorry. So hopefully you've maybe cottoned onto the fact that I'm definitely not going to be using commercial probiotics to have this effect. It's not that I'm an anti-probiotica, not like an anti-vaxxer, but it's just that we're trying to replenish a whole ecosystem of diverse gut bacteria that have been ablated from chemotherapy. And so a probiotic made of just one or two types of gut bacteria is just not going to be large enough or impactful enough to change or restore these people's gut bacteria. So I've decided to take a slightly different and less glamorous approach, and I'm going to be using fecal microbiome transplantation, or FMT, or a poo transplant. A few nods in the audience, that's quite good. I'm impressed to hear that some people have heard of this. That's good.
2 years ago, is his name Tarrant?
It's a good question. I don't know a Taras. Okay.
It was his topic a couple of years ago.
Yes, you're right. Yes,
The
it was.
relationship between the brain cells and your bowel.
Yes, well, absolutely. I mean, there have been some really interesting cases with fecal transplants where, for example, we've been able, well, not we, but others have been able to transfer certain phenotypes. So depression has been shown to be transferred in a fecal transplant, only in mice at this stage. But there have even been examples of an obese mother giving her daughter a fecal transplant, and that daughter then becoming obese. So there are certainly some interesting case studies in this space, but I want to emphasise these are case studies where it's an N of 1 usually, so we can't really draw any major conclusions from these studies. But for those of you in the audience who are maybe thinking, what on earth is a poo transplant? It's basically where we collect stool or poo from a healthy donor, and we then use that poo to give it to someone in order to change or improve the composition of their gut microbiome. And I'm very sorry for people who are hopefully still not eating. What I want to emphasise before your imaginations start to run a little bit wild is that we don't just go up to anyone on the street and say, hey, can I have a poo? And then, I don't know, feed it to someone else. You know, it's a very heavily regulated space, and we have to do a lot of, crossing of T's and dotting of I's to make sure that we follow very strict protocols in terms of who our donors are, how the stool is processed, and also how it's administered. And so to help us navigate this very complex regulatory landscape, we've actually partnered up with Australia's leading stool bank, which is actually located right here in Adelaide, just in Theberton. And for those of you that are wondering what a stool bank is, it's exactly like a blood bank, you know, where we all hopefully go to donate blood, but instead of donating blood, you donate a little bit of poo. And so usually these stool banks are filled with the poo of very eager, slightly poor but healthy uni students who are more than happy to poo on demand for a little bit of money. But luckily they are still very healthy, and we do very strict health assessments of all of the donors that come through the stool bank. And Sam Costello, who's actually the director of the stool bank, who was meant to be here with us tonight but unfortunately couldn't be here, he's really revolutionised and pioneered the use of fecal transplant products in South Australia, and actually he's now pushing into Australasia. And he's really used fecal transplants very effectively in the treatment of inflammatory bowel disease, which is a condition of the gut which is typically associated with episodes of very severe and painful diarrhoea. Now, what he was able to show was that by giving these people a fecal transplant from these donors, he was able to induce or elongate the amount of time between these episodes of diarrhoea. And actually, in some people, he was able to show complete remission, which is a pretty profound finding, because anyone that's familiar with IBD would know that it's a very challenging condition to manage, and it's pretty much impossible to cure, really. And so what Sam's findings really emphasise is that fecal microbiome transplantation is a pretty major advance in our ability to manipulate people's gut bacteria and also to restore the composition of their gut bacteria. But like I've been saying, you know, the use of donor faeces, in Sam's situation, it's unavoidable. These people have inflammatory bowel disease, they've already got a disrupted microbiome. But what we're starting to understand is that different donors induce different responses. I mean, like I said, we've all got different gut bacteria. So a fecal transplant prepared by different people is going to have a different effect. And this has sort of led to this phenomenon of the super pooer, which I think is quite funny. And I get a lot of questions about it, particularly in mainstream media articles and things like that, because people are just fascinated by these super pooers. And basically, these are people who their poo, when it's used for a fecal transplant, has a particularly beneficial response. But unfortunately, we have no idea what makes these people super pooers. But what we do know is that people tend to respond better to their own gut bacteria compared to those of others. And so in oncology, we have a really unique advantage because we can actually collect people's poo before they start their chemotherapy. We can bank it at the stool bank, keep it in a really cold freezer so all the bacteria are happy and healthy. And we can then re-administer that to people when they've finished their chemotherapy in order to restore their gut bacteria. And this is exactly what we're hoping to do in this study. And we're hoping that by restoring their gut bacteria, we can prevent the complications that are associated with damage to the microbiome, so things like diarrhoea, infection, graft versus host disease. But what we also hope to be able to do is minimise the use of antibiotics in these people, which we know are associated with problems like resistance. And also, we just want to keep these people or make the lives of these people a little bit better while they're going through something extremely traumatic and challenging. And ultimately, the goal is to make sure that these people don't just survive, but that they truly thrive. And so, with that said, I'm going to wrap it up there and throw it over to you guys.
Thank you so much, Hannah. Okay, so we do have some time to ask a few more questions while she has a drink, because she's a bit parched at the moment. So does anyone else have any questions for Hannah tonight? Yep.
With the super poor idea, and like, I was just thinking, is there much literature on like rejection, kind of like when people have tissue grafts? If you have a fecal, there much chance of like the differences in the outcomes? Is that because of tolerance, immune tolerance?
I think that's a really, really good question. And it's something that I'm hoping to slightly probe in this space of graft versus host disease, because GVHD, or graft versus host disease, is a disease where it occurs from a blood or a stem cell transplant from a donor. And so there's this level of rejection. And so I really want to compare how someone responds to their own fecal microbiome transplantation compared to that of others, and whether there is more tolerance to our own. It's something that we are able to perhaps conclude or at least hypothesize from the literature at the moment, but it's really difficult to tease apart. And actually, it's a great question, because I've put it in a recent grant application to answer that exact question. So watch this space. Hopefully I get funded, and then in a few years I'll give you the answer.
Given that we've all got unique microbiomes and whatever, and we respond differently to other people's fecal transplants, are you doing that in isolation, or as part of like changing people's diets and getting them into meditation for stress and stuff like that, or is it just isolating the effects of the fecal transplant?
I mean, I think you raise a really good point that it's all well and good to support people's gut bacteria, but if they're still stressed, or the stimulus or the factor that's damaging it is still present, then it's kind of, it's silly. So we're also looking at ways in which to support the gut microbiome in addition to fecal transplant, and you're exactly right. We're looking at things like exercise, high fiber diets, Mediterranean diets, and the space, or the exercise space at the moment is quite interesting. I mean, yeah, they're showing things like yoga have positive influences on the composition of the microbiome, and I guess the way in which I see it is that it's got to be a bit of everything. I think, for people going through chemotherapy, the significance or the magnitude of the change that we're seeing in their gut microbiome is pretty profound. So I think it's probably unlikely that diet and exercise will be enough to help them rebound properly. So I think it's probably got to be a combination of these things for sure.
I do have a question myself, if I can.
Yeah.
From the second lot, the questions we did in the beginning, from an environmental factor and chemical type things, obviously that's a huge effect. Can you tell us a little bit more about some of the types that are standard that really we need to probably be a little bit more aware of, even with our kids and stuff like that?
Yeah, absolutely. I mean, I'm certainly not a microbiologist in that sense, but I can certainly give you some pieces of information to help you with that one. There's a really interesting study at the moment that's happening at the university that I was at in the Netherlands, in a town called Groningen. And they've sort of, I guess, leveraged on the fact that suddenly, because of COVID, we're all very, hygiene conscious. And I never used to use hand sanitizer, and now I sanitize every time I walk into Coles. And I also get angry at people that don't sanitize. And so they're trying to look at the gut bacteria of people in a post-COVID world to see if this sort of hypervigilance on sanitisation has an influence on the types of bacteria that are found in our gut. And my inkling is that it probably will have quite a profound effect. But what that means in terms of, risk of disease and the flow-on effects, it's a little bit undetermined at this point. But I think it also goes back to that point that I raised in the first half of the presentation, which was that people with siblings and pets, they tend to have a more diverse microbiome, probably from being exposed to a few more pathogens and bacteria throughout their development. So I think, from a gut microbiome perspective, I would certainly be in favour of, kids eating a little bit of dirt, but maybe don't quote me on it.
Do we have one more question? Excellent, up the back.
Thank you, Hannah. That was a terrific talk.
Thanks, Julie.
I was thinking about hearing of sniffer dogs and being able to detect different diseases by smell, dogs being able to train to do this. And is that somehow related to people's microbiome and what they eat? And is there any kind of potential for those volatile products of
Poo.
both what we eat and how we, our emotional well-being and whatever, to kind of be more of a final product than just looking at the microscope and seeing what bugs are there?
Absolutely. I mean, I think I was actually going to touch on this point in this talk tonight, but unfortunately the list of things I wanted to discuss just got a little bit too long. So what you're alluding to is what we call an olfactory signature of the gut microbiome, which is basically a fancy word for how it smells. And what we know is that the olfactory signature of an individual's poo or even their breath indicates or is reflective of the type of bacteria that are found in their gut. And so there have actually been some studies in the field of cancer therapy in people going through, I think it was radiotherapy for a gynecological cancer. And this was only done in, I think it was about 20 people. But what they were able to show, it wasn't using dogs, but it was using a special machine called an e-nose, an electronic nose, which I also think is hilarious because I picture this big nose, but it doesn't look like that. And basically this special machine is able to detect this olfactory signature of the stool, and it's able to create a unique spectral signature, a squiggly line basically. And what they were able to show is that this olfactory signature was different in people that went on to develop complications due to radiotherapy versus those that didn't. And so this is very much in support of this idea that our gut microbiome and our gut bacteria predict how we're going to respond to therapy. And I think it's a really interesting way of looking at the microbiome, because the way in which we do it at the moment is by using a genetic sequencing approach. And this takes a few days. You have to get the stool sample, extract the DNA, amplify up the region of interest, and then run it through a special sequencing machine that tells you all the different types of bacteria. But the beauty of an e-nose is that it gives you this one readout in a matter of minutes. And so the translational capacity of this work or of this approach is much higher than what it is for bacterial sequencing. So again, you guys are all over it. You should be writing my grants, because that was in my grant as well. So come on board and start writing my grants for me.
Do we have
Julie.
any other questions? One last question maybe for the night? Gee, one more? Absolutely.
Jack, all over it.
Would part of the toxicity of chemotherapy be some bacteria, when you treat them with antibiotics and when they die, they act like suicide bombers, and when their cells rupture, they let out things like LPS? And I was wondering if toxic shock syndrome is part of that.
Absolutely, yeah. So the work that I did in my PhD was looking at the role of a receptor called toll-like receptor 4, which binds to or is the receptor for LPS, which is produced by bad bacteria or damaged or dying bacteria. And what we were able to show was if that we genetically deleted this receptor from a special line of mice, we were able to really minimise the intensity of the toxicity that was associated with chemotherapy. So that allowed us to conclude that the effects or the contribution of the microbiome to treatment toxicity was mediated through toll-like receptors. And actually, Georgette, in the crowd, I can see her getting excited. She's carried on some of this work for her honours project this year, because we were really interested in whether this phenomenon occurred because of the TLRs or toll-like receptors that are expressed on immune cells or those that are expressed in the gut. And so Georgette's been really integral in helping us understand which subset of cells is responsible for this finding. So thanks, Georgette. It's been a tough year, but you've made it. And Jackie. All right.
Okay. Do we have another one, maybe?
If anyone wants to come up afterwards, maybe talk.
Maybe, yeah, if you'd like to have a chat a little bit later.
More than welcome.
I just want to say thank you for coming tonight and giving our speech. And Ellen's just going to present you with a bottle of something delicious for you to have.
Great.
So can we all give Hannah a round of applause and So I would encourage you all to, obviously, if you do want to grab Hannah and ask her another question, or maybe buy her a drink at the bar, bar's still open. And yeah, make yourself welcome over there, guys. Thank you so much. Thanks, guys. I hope you enjoyed it all.